When the human genome was first sequenced, scientists were shocked to learn that there's only a few thousand genes in the genome. In fact, all the world's top geneticists had kept a betting pool. Yes, they bet real money. After they sequenced the human genome, was going to be announced how many genes there were, and the geneticists that came closest to the answer would get all the money. And they all guessed too high. Every single person who was in the middle of that gene sequencing stuff actually had no idea how few genes the human genome had. The final tally was about 22,000. I did an episode a couple of months ago, The Incredible Shrinking Human Genome, where they recounted it, maybe 19,500, which is not a lot of protein coding genes when you consider that the human genome produces over probably more than that, 100,000 unique proteins. In fact, different cell types will produce similar proteins as another cell type, but there'll be pieces that are different because they can rearrange the parts of the gene as the protein is getting made. One of my first ever biblical genetics episodes was Slicing and Dicing the Human Genome. In fact, there's an article on creation.com, Slicing and Dicing the Human Genome. Also, it deals with the incredibly sophisticated and amazing way that genes are rearranged before the proteins are made. Therefore, you can have a relatively small number of protein coding genes produce a fantastically huge number of unique proteins. Splicing and dicing, compactness of the genome, it is all amazing, it is all ingenious, it is wonderfully engineered because the God of creation is amazing. Well, hey, I'm Dr. Rob. I'm actually coming to you today from my new office. I just got moved across the hall. I had to move all my books. I had to move all my pictures. I had to move all my fossils. And so I'm just setting up here. It's a Saturday afternoon. No one else is around. I've been listening to some podcasts, listening to some music. And I just decided it's time to stop because I need to give you a new episode. And this episode is about something brand new. No, it's not the number of genes we thought were in the genome. It's the number of genes we now know are in the genome. Because my incredible shrinking genome episode has been completely contradicted by brand new information. I give you the Dark Proteome. So if you were to look for a gene, if I were to give you a piece of DNA, here's the code on a piece of paper, computer screen, it says like ACGCTTTACGGC. It's just a lot of letters. Where are the genes in there? How do you find them? Well, one thing you can do is look at a protein, sequence the protein. And then once you know which three-letter combination produces each amino acid in the protein, you say, well, if that's this amino acid, then it's got to be those three letters, except some amino acids have more than one letter set. Some of them have 4, 5, or 6. So you have to do a little guessing. But you can say, okay, well, if this is that amino acid, it's those three letters probably, maybe this letter could be different. And you can line up all your amino acids, all the possible codes, and then search through your DNA for something that matches. That can be done. So another thing to do is to look upstream of the gene. Upstream of a gene is called the untranslated region. When the ribosome makes a copy of the DNA in the form of RNA, it will not just copy the protein coding region, it will copy upstream also. Up there's places where the ribosome attaches. There's different things, signaling factors that can turn a gene on or turn a gene off or ramp it up or ramp it down. All sorts of things can interact with the DNA upstream of that and then also interact with the RNA upstream of that. So not only can the parts of the gene be rearranged to make different proteins, there's parts of our genes, not bacteria, but higher organisms that are in the middle of the gene that have to be cut out and thrown away. They're called introns or intervening spaces. The parts that code for protein are called exons, and the introns are in between the exons. The introns have to be cut out, the exons joined together. And when they're joined together, you get what's called the messenger RNA. And the messenger RNA can be translated into protein in this very complex thing called a ribosome. Okay, so we have introns, we have exons, we have genes, we have lots of complexity here. How are we going to count the number of genes in the human genome? Well, we could look for things that are classically associated with genes, like almost all protein coding genes starts with the letter ATG. That's the codon or the three-letter code for methionine. So most proteins start with methionine. It might get chopped off later because lots of protein modification happens, but most proteins start with ATG. You start with an ATG and we go 3 letters at a time, because each 3 letters codes for one amino acid in the protein. 3 letters at a time. You go long enough, you'll run into what's called a stop codon. UGA is one of the 3 classic stop codons. Now, one of the stop codons in the human genome can also code for the amino acid selenocysteine. So we actually have 21 amino acids with 21 different transfer RNAs, but it's another subject for another day. Point is, start, stop, and everything in between gives you your protein, generally. Now, most genes, they might be 300 to maybe 2,000 amino acids long. Some are shorter, some are longer. But the average gene, let's say it's like 1,000 amino acids long. So it's about 1,000 codons, or about 3,000 letters of DNA to code for a single gene. Yeah, some are much longer than that, but in general, about that length. A gene, though, will probably have maybe 20,000 letters. There's going to be the upstream and the downstream regions that aren't going to be translated into the protein, the untranslated regions. There's going to be a lot of introns in there. In fact, most genes, there's more intron than there is exon. Some of the introns are longer than the gene product itself. So you have to cut out the introns and join the exons together. So you have about 20,000 letters-ish to make a 1,000 amino acid protein. That's the general size of things. And so knowing that, geneticists have tried to look for long stretches of DNA between a start and a stop. Because if you have a long one, you're almost certainly in a coding region. Why? Because if you have just random DNA, you can find an A. Sure. That's 1 out of, let's say, 30% of the time, if you're looking at a random letter, there's an A there. You might find a T. Okay, it's also 30%. A G is about 20%. So knowing those frequencies, you can calculate the frequency of an ATG. Fine. But then the next 3 letters, and next, because there's 3 stop codons and there's only 64 possible codons, about 1 out of every 23 letter combinations is a stop codon. So in random DNA or non-coding DNA, there's a lot of stop codons. Even if you find an ATG, you maybe have to go down 20, maybe 30, maybe 5, some range, but you don't have to go very far before you find a stop codon. So in coding regions, you got long stretches with no stop codons, as long as you're in frame. As long as you have these 3 letters where you start with, the next 3. If there's a mutation in there that like deletes a single letter or adds 2 letters, you have what's called a frame shift mutation. And a frame shift usually destroys the protein, because as soon as you shift out of frame, you're into random territory, and it's only going to be probably maybe 20 codons later before you get a stop codon. So a lot of proteins are truncated by random single or 2 insertions or deletions of letters. All right, I just threw a lot of terms at you. We have introns of exons, we have codons, we have in frame, we have an upstream and a downstream untranslated region. All those things are hallmarks of a gene, and you can write computer programs to find them in genomes. You can actually scan for them by eye. There's one point in time where I could look at a stretch of DNA and I could actually go from the DNA and code it into a protein. In fact, in my green fluorescent protein family of proteins that I was working with in graduate school, I would know where in the protein I was just based on the letters of the DNA that I was staring at. I had spent so much time staring at that I could almost translate it on the fly. It was really weird having that much detail about that one little thing. But that's just one little thing. We want to know how many genes are in the human genome. Turns out to be extremely difficult. And one reason for that is there's a size limit. Really tiny proteins are lost in the noise, because in a random stretch of DNA, you might expect to find every once in a while, given 1 billion letters of random DNA, you're going to find what's called an open reading frame. There's another term, an ORF, an open reading frame. That is, start codon, go boom, 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 down to DNA. Every once in a while, you can find one that's really long and has no stop codons in it at random. So is that really a protein, or is that just a random stretch of DNA that just happens to have no stops? Or is it a deactivated gene, a broken gene? Or is it a highly repetitive section of the DNA that just doesn't have any G's in it? Therefore, there's not going to be a stop codon. That's hard stuff to know. So this new paper has come out. They actually did an editorial on it in Science magazine last week, but it's not published yet. The paper is still on bioRxiv on a preprint server. I thought that was really weird. I mean, have we gotten to the point where non-published papers on preprint servers are so important that we actually didn't talk about them as if they're finished. Well, that's what they did in science. It was Elizabeth Pennisi wrote this essay, this 2-page essay on this new idea. And I think the reason that she did that was because it was so groundbreaking. What they did was they took the ribosomes and they froze them in place as they were grabbing onto an RNA. And then they sequenced all of the RNAs that were associated with a ribosome. In other words, the thing that was doing the translating into a protein. And they were shocked. They found, no joke, tens of thousands of new genes. Tens of thousands. And these were what we thought were junk DNA. So now junk DNA is taking another hit. Yes, once again, a new function of what we thought was non-functional DNA. Most of these new proteins are really small, so they were lost in the noise. There are massive databases of all of the proteins made by different organisms. In fact, different tissue types in different organisms. In fact, different cell types in the tissues of different organisms. There are massive databases of all the RNAs that are produced by organisms, by tissues, and by cell types. And yet, even despite that, we didn't find these proteins because of the tiny sizes. And some of them are really repetitive. It's just the same thing again and again and again and again, like the antifreeze protein in the ocean pout, which is a really simple protein. It's just very repetitive. But because of all that repetitive protein, when you add that to a bloodstream, you can't get ice crystals to form in the bloodstream. So some real simple proteins are really important, including this. In fact, these researchers have already identified a particular one of these little proteins that they thought was just junk that's strongly associated with one form of childhood cancer. So already, and this is really rare, that a brand new field comes up and they instantly find an application. Now, let's find a drug to target that mutated protein in those children that they didn't even know was a factor. So that is crazy, cool, and fun. To review, initially there were not enough protein coding genes in the genome. And then we shrunk a little more. And now we've just taken it, at least doubled, maybe even trebled the number of genes in the genome. It's just that the genes we couldn't find because they're small and thought to be in the junk DNA. Okay, let's break into a little personal story here. When I was in graduate school, I was working on the green fluorescent protein family of proteins. These are in corals, they're in jellyfish. The bioluminescent jellyfish Echoria victoria was the first time anyone had found a green fluorescent protein. It's really weird because this jellyfish glows green at night. It produces green light. But when they figured out the bioluminescence system, it actually produces blue light. But there's an accessory protein that grabs the blue light and changes the wavelength and refluoresces it at a slightly lower energy, a longer wavelength. It comes out as green light. So the green fluorescent proteins. Now, at the time when I started, that was the only green fluorescent protein known. My team, we pulled green fluorescent proteins out of corals, brand new green fluorescent proteins. They had the same function as the other, same wavelength as the other, but it's a completely different sequence. And then we targeted the red fluorescent protein. And that when I was scooped, I actually had cloned a red fluorescent protein. And I'm sitting there in my laboratory saying, wow, I just made millions of dollars. Well, I didn't, because that day a paper had come out, red fluorescent protein by Mikhail Matz and his team in Moscow. It's really kind of funny because I think they used the same soft coral that I used. I went down to my aquarium store in Miami and found this bright red little disc. And I took it and I sliced a piece out of it like a piece of pie and put it back in my aquarium. And it healed up and kept growing. And I took that and extracted the RNA out of that. And I made red fluorescent bacteria from that. Well, they had done the same thing, but they had about a 2-year head start on me, and they had a lot of money and a big team. They worked really hard on something for years, and I did it in 48 hours. But that's another story. You can read my paper to find out how we did that. Anyway, we're working on these green fluorescent proteins. We're mutating them, changing their color, making them brighter, making them dimmer, putting them into fish, next to specific genes in the fish so that they would turn on when the gene turned on. It was a lot of amazing stuff. But I wanted the gene. What we did, we took a shortcut. We wanted to clone this gene without going through all the laborious steps of sequencing the entire gene. So what we reasoned was if we just take a coral and chop it up and then extract all the RNA out of the coral, every type of RNA possible, and then converted that RNA into DNA, and then take all that random DNA and shove it into bacteria, let the bacteria pick up random pieces of DNA. And if we plate that bacteria and let it grow, any bacteria that had absorbed a messenger RNA from a green fluorescent protein would be green. And it worked. It was amazing. In fact, the University of Miami's patent office funded my research because we had this brand new idea and this brand new way of applying this technology, and it worked. And we sequenced and we cloned a whole bunch of different colors of different fluorescent protein genes. Okay. That was a lot of fun. But in the middle of all that, I wanted to know the gene. How is it different than the jellyfish gene? See, when we took the RNA out, we did that because bacteria can't splice introns. Bacterial genes don't have introns. Higher organisms have introns. If you want to put the DNA in a bacteria, it wouldn't have done anything if the introns were there, which is why we're targeting the RNA, because some of the RNA would be messenger RNA. Get it? Kind of cool, huh? Well, once we had that, I said, okay, I developed what's called a primer. That's a little piece of DNA that sticks onto a longer piece of DNA. And in the polymerase chain reaction, you have two primers, one for the beginning, one for the end, and it will amplify. It'll make copies of the DNA between your two primers. And you can make many, many copies of DNA. It's something that is used all the time in biotechnology, PCR, or the polymerase chain reaction. But I didn't want to do that. I wanted to go into the unknown area, which means I couldn't have an end primer, just the beginning primer. And I did something called long distance PCR, which I just made a primer for a space that I knew, and I let it run out into the unknown using the original coral DNA, meaning the DNA of the genome of the coral, which has introns. And sure enough, I got to sequence into the introns, and then I would make a primer for the next spot and sequence a little deeper, make another primer until I got to an area that I recognized because it was in my original clone protein. Cut out the introns and join the exons together. Well, I just sequenced through the intron into the next exon. And I found the gene sequence. That was a lot of work. It took a lot of money also. Back then, DNA sequencing, like year 2000, was expensive. It was laborious. It was slow. But I found a company that had just started up that they would sequence anything I had PCR'd for $20 a sample. Oh man, it saved me so much time. Because back in the day, in the sequencing machine we had, I had to pour a gel between two pieces of glass that are separated just a little bit. This giant piece of glass. It was probably a meter tall or more, 3 feet by 4 feet by 2 feet. I don't know. It was this massive 2 sheets of glass. And I had to pour basically Jell-O, but an agarose gel into it with no air bubbles. Oh boy. And on the top, we put a comb. And so when you pull the comb out, there'll be little nooks in the top of the gel. You put that in the machine, you could load your DNA into it, and it would run an electricity gradient through that gel, and the DNA would start migrating down to the bottom. And of course, the smaller piece of DNA would go faster than the larger pieces of DNA, and the bottom was a laser. And the laser would read out different colors because it's something like when you were making copies of your DNA, you added a special chemical, and A, C, G, and T would each be labeled with a different fluorescent molecule. And so when it got to the end, if the reaction stopped with an A, it would be one color. It had to stop with a G, it would be a different color. And the laser would go ding, 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 and it say A, C, G, C, G, C, G, A, and all your different columns. But man, that was hard. It was so much better when I could just send it to somebody else and let that laboratory do it. Today, sequence technologies are almost trivial. I'm kind of laughing at the Stone Age technologies I was using, but man, back then that was high tech and amazing. And I sequenced my genes. And I saw that there's an untranslated region, the beginning and the end. And my genes, the two of them that I sequenced, one of them had 4 introns. One of them only had 2 introns. In fact, the 2 introns in the red fluorescent protein were in the same place as the first 2 introns in the green fluorescent protein, but the red fluorescent protein didn't have the other 2 introns, and one of the introns in the red fluorescent protein was massive. So that's the nature of DNA. Now, you want to take that, and you want to find the genes in a genome, you can't do it. Because just because something looks like a gene doesn't mean it makes a protein. In fact, there are more non-coding genes. start codons, stop codons, introns that can be alternatively spliced that actually don't make protein than there are protein coding genes. Now, those non-coding genes aren't junk. They make RNAs. The RNAs are where all the work is being done in the cell. Incredibly important stuff. But if you want to find the genes, you gotta work really hard. That's my point. It's a lot of hard work. So I can't quite fault the geneticists for having missed half of the genes in the genome for the last 20 years. Then again, it was the assumption that junk DNA exists that caused them to not bother to look. They didn't expect to find thousands of little teeny genes. They didn't. And they didn't expect to find thousands of little teeny genes that are active in the cell, that are functional, that actually do things. So this new discovery has the potential of completely revolutionizing our understanding of the genome, how the genome works. It is going to have an impact on medicine. It is just a cool, interesting, fantastic, and fun new discovery that I just wanted to share with you. Now, there's an article on creation.com. You can go find it on this very topic. What did I call it? I think I just called it the Dark Proteome. It'll be there on creation.com for you to read and enjoy. But in the meantime, thank you for listening. I really appreciate it. Thank you all so much for my sponsors. You guys are awesome. There's some links in the show notes if you would like to help sponsor the show, just to keep me on the air, because I'm actually going to go home now. I'm going to start editing this and probably be up late doing that. Tomorrow, I'm speaking at 2 big churches in Atlanta. That's going to be a long day. I got to leave at like 6 in the morning. I'm not going to get back to probably 10 o'clock at night. I get Monday off, and then Tuesday I'm back here in the office, writing articles, editing videos, writing books, and things like that. We work really hard here at CMI, and this is nonstop, go to produce information for you, hopefully to encourage you, to edify you, to exhort you a little bit. If you don't actually believe that God exists, or if you don't believe that God is the creator, I would encourage you to take a second look, because the genome itself shows us there must be a creator, a higher power who took his mind and organized matter in such a way to allow life to exist. And that includes programming the information in the genome, which is continually shucking us.